Topic 14a, DNA replication. DNA replication is the process of beginning with a single double-stranded molecule and ending with two identical copies. DNA molecules are replicated by denaturing an existing double strand into single strands, and the resulting single strands act as templates for the synthesis of new single strands. This produces two partially new and partially old double-stranded molecules. This process is catalyzed by several enzymes. Let's go through it stepwise. The initial step is to denature the double-stranded molecule. And first, we'll focus on the synthesis of just one of the strands. The primary enzyme that will make new DNA is called DNA polymerase. But DNA polymerase can't just start. It requires an existing 3' hydroxyl to begin synthesis. For this reason, the next step is the addition of a short RNA primer by the enzyme primase. Notice that the primer was synthesized from the 5' to the 3' direction. DNA synthesis enzymes can only work in one direction, and that's 5' to 3'. Once the RNA primer is in place, the 3' end provides a hydroxyl group that the enzyme DNA polymerase can add a nucleotide onto. This enzyme will continue to synthesize by adding new nucleotides as long as there is template available. DNA is double-stranded, and the strands align in opposite orientation. That means that synthesis of the two opposing strands must occur in opposite directions. Now, as we move down the template strands, the two strands will continue to denature and accommodate synthesis. One of the strands will continuously open in the direction that the DNA polymerase is progressing. This is called leading strand synthesis. The other strand cannot be synthesized in the same manner because DNA polymerase can only make new DNA 5' to 3'. So on that strand, a new primer is made, and DNA polymerase will synthesize DNA in small stretches. This fragmented type of DNA production is called lagging strand synthesis. One of the final steps is when a different DNA polymerase will remove RNA primers and replace them with DNA. To review, when DNA is synthesized, an existing double-stranded molecule is denatured, and each single strand acts as a template for the production of a new double strand. This process is enzymatically catalyzed. DNA synthesis can only proceed in one direction.